Hello guys, welcome back to my lesson. So today we're going to do questions under the topic of DNA code of life. So let's look at the first set of questions. It says that the diagram below represents transcription during protein synthesis. So let's look at the diagram. The diagram, it looks like it's showing, yes, a transcription, but this looks like a DNA molecule only with the nitrogenous pairing, right? But if you look at the diagram very well, they have written mRNA also. So you just have to take note of that. I think the examiner was just trying to confuse us here. So the first question says that name the part of the cell where this process occurs. This process is transcription and it takes place in a cell, but specifically in nucleus. The answer is nucleus. Identify sugar X. Sugar X is deoxyribose sugar because they've written DNA down there. And nitrogenous base Y, I remember the other side is written mRNA, not DNA. So it means that Y is going to be uracil. They say now that tabulate two differences between transcription and DNA replication. So let's look at the process of DNA replication and the process of transcription. So the first difference that we can see here is that only one strand acts as a template in transcription and both strands act as templates in DNA replication. Another difference is that mRNA molecule is formed and then in transcription and in DNA replication, two identical DNA molecules are formed. So now let's look at the last one. The last one is that DNA unwinds and unzips partially. And in DNA replication, DNA unwinds and unzips completely. So in DNA replication, the reason why it unwinds and unzips completely is because one DNA molecule is going to have one strand that is new, and then the other one that was the part of the first DNA molecule that we had. So let's look at the next set of questions. We have a mutation occurred on a section of an mRNA molecule as shown below. Let's look at the original sequence. It has AUG, GGA, AUA, CCG, CCA, GGA. And the mutated sequence, it's the same as the original, but the difference is instead of CCG, there's CUG. So they say name the type of mutation. The mutation is gene mutation. Why? Because it is a change in the sequence of nitrogenous bases. If it was the change in the sequence of chromosomes, it was going to be chromosomal mutation. If maybe in meiosis, one chromosome refused to, did not separate, so it was going to be chromosomal mutation. So let's look at the questions. Another question says give a reason for your answer because there's a change in the sequence of nitrogenous bases. So let's look at the next question. It says the table below shows some mRNA codons and the amino acids that they code for. Though you don't have to know amino acids by heart, they'll always give you these amino acids. So this table has mRNA, which are codons, and then the question says state the number of different amino acids coded for by the original sequence of the mRNA molecule given above. So they want the number of the amino acids that will code for the original sequence. Let's look at the first base triplets. We have AUG. Let's look for AUG, methionine there. We have GGA, glycine. We have AUA, isoleucine. We have CCG, proline. We have CCA. It's also proline, and then GGA, glycine. Oh, GAA is glutamic acid. So we have 5 of them because proline, we don't count it. We don't count it twice. It's repeating itself. And then the next question says that give the anticodon on the tRNA molecule that carries the amino acid isoleucine. Now we go to isoleucine and then we look at the base triplet given for isoleucine is AUA, which is for codons, right? mRNA. Now, when you're answering this type of questions, you're going to write the sequence of DNA, and then you're going to write mRNA, and then you're going to write tRNA. You're going to use the sequence, and then you look at the base triplets that you're given. You're given AUA for what? For mRNA, you substitute it on mRNA, AUA, and then for tRNA, A pairs with what? We're going down, A pairs with U, because we are in mRNA. U pairs with A, and then A pairs with U. So the answer is UAU. So you use this sequence whenever they ask you this type of questions. So now imagine if they have given you DNA to say that it's TCA. So what you're going to do, you're going to use the same sequence, but you would have to, if maybe they asked for the tRNA base triplets, you are going to write the mRNA first, going down. T pairs with, it pairs with A, C pairs with G, A pairs with U. And then before you can get your tRNA answer, you have to move to mRNA. Now we have AGU for mRNA. And then for tRNA, A pairs with U, C pairs with G, U pairs with A. The answer is going to be UCA. For an example, if you're given base triplets for tRNA, you were going to get the ones for mRNA first, and then they wanted DNA, and then you move to DNA after moving from mRNA. You don't move from DNA to tRNA straight. So now let's look at the next question. Says that use the information in the table to describe the effect of the mutation on the protein formed. This is simple. You just tell them that the codon CCG changed to CUG. The tRNA sequence has changed. The amino acid proline was replaced with leucine. This resulted in different protein, and that is all. You're going to get your full mark. Let's look at the last set of questions. We have the diagram below represents the DNA profile of 3 children and their parents. Only 2 of the children are their biological children and 1 is adopted. So how you answer this type of a question, you look at the bands of the mother, the DNA bands, which are those black lines, horizontal lines shaded with black, are the ones for the mother and the father. And you look at the children's bands. You identify, you need to look for their biological children, right? You look at the mother, the first band matches with that of Hala, the second one matches with that of Leo, the third one matches with that of Leo, and then the fourth one matches with no one. The fifth one matches with Priya, and the last one matches with that of Hala and Leo. So and then you look at the father. The first band matches with Hala and Leo. The second one matches with Priya. The third one matches with Hala and Leo, and the last one matches with that of Leo. So you would, if you look at the bands in the mother and the father, they matches with that of Hala and Leo, not Priya. So the first question says identify the 2 biological children. The answer is Hala and Leo, because all the DNA bands from Hala and Leo match with that of the mother and the father. That's what you're going to explain in 2.5.2. Let's look at the last question. Says that state the 3 other uses of DNA profiling. You're going to write tracing missing persons, identification of genetic disorders, identification of suspects in a crime, matching tissue for organ transplant, identifying dead persons. Thank you so much for listening. Don't forget to subscribe to the channel. Thank you.